Welcome back to our protein journey. Today we will continue to explore protein structures, from their different levels of organizations to the intricate bonds that holds them together. Emphasis will also be placed on the dynamic nature of globular proteins, as well as that of fibrous proteins. In our previous video, we would have learned that, when hundreds of amino acids join together, they form polypeptide chains, which therefore gives rise to a protein. But do you believe these proteins can have different structures? Correct, yes they can. There are, in fact, 4 different structure, or levels of proteins, that you need to be able to describe. The first level of protein structure is called the primary structure. The primary structure is simply the sequence of amino acids in a polypeptide chain. In other words, when more and more amino acids join and establish peptide bonds, it forms the primary structure. So, I am showing you here an image on the left, exemplifying a prime example of what we are trying to say. Each 3-letter abbreviation, or codons as we call them, represents one amino acid. As such, it is just a sequence of these amino acids that makes up the primary structure. It is important to note that changes in amino acid sequence, even of a single amino acid, can disrupt normal protein function entirely and result in disease. Coming up, we are going to look at the secondary structure of proteins. In the primary structure of proteins, the amino acids are linked by peptide bonds, and the peptide bond has an oxygen attached with a carbon, and the nitrogen attached to a hydrogen, as can be seen. Both oxygen and hydrogen has the ability to form hydrogen bonds. And this results in folding of the polypeptide chain into either an alpha helix or a beta-pleated sheet. Let us talk about the alpha helix. Alpha helix is the most common type of secondary structure of protein, and this structure of protein attains a spiral shape, such that the side chain of the amino acid remains outwards. The alpha helix is stabilized by hydrogen bonding. This hydrogen bond is formed between the hydrogen of nitrogen and the oxygen attached with the carbon. Another type of secondary structure is called the beta-pleated sheet. In this case, the polypeptide chain folds into a flat or sheet-like structure. Again, hydrogen bonds between amino acids hold the shape in place. The further folding of the secondary structure gives rise to the tertiary structure of proteins. Therefore, the tertiary structure represents the overall folding and coiling of the peptide chains. It is the tertiary structure of protein that gives a geometric shape to the protein. That can be fibrous or globular shape. The tertiary structure may have other bonds such as disulfide bonds, hydrophobic interactions, and ionic bonds. We can use biological enzyme as our example, as they contain these bonds. There are some proteins which consists of 2 or more polypeptide chains. These polypeptide chains are referred to as the subunits. Hence, the quaternary structure is the special arrangement of these subunits with respect to each other in the protein structure. Depending upon the molecular shape of proteins, they can be classified as fibrous proteins and globular proteins. Let us speak some more about each to gain some insights. Fibrous proteins are formed when polypeptide chains run parallel to each other and are linked together to form fibers. The molecules of these chains are held together by hydrogen and disulfide bonds at many regions. These proteins are also insoluble in water. Some common examples of fibrous proteins are collagen and keratin. On the other hand, globular proteins are formed due to the folding around of polypeptide chains. This gives us a spherical shape to the globular proteins. These proteins are soluble in water. A few examples of globular proteins are insulin and albumin. As you can see, the structure of proteins are quite complex. So that's a wrap. I hope this video was very helpful. Here is an image to reiterate what we have discussed.